Nivea people to save me. I'm gonna be outside all day. I want you all to spray me. I'm gonna close my eyes and spin.
Okay. Hey,
all over here. Get my bald spot. There you go. Oh, this is good. First, we're gonna set up like a Netherlandish still life, a tabletop with the nice linen. Patrick Foss picked it out, yes. And then we're gonna cut off little pieces of every little organism, you know, some meat, vegetable. We've got all 5 phylums, fungus, bacteria. I bought some yogurt. It's got bacillus. And like protists. I think we got some swamp water in the ice cooler with protists in it. So we're gonna mix them all together in the blender. Other things you can do with it is just inject it into living things and see what happens. I guess we'll just figure out if people want to do it. It's up to them. This blender came with something called a power stick. It's like a jammer, a kind of sex blender jammer thing.
Ik ben Laura. Ik ga een stilleven maken.
Wat heb je gepakt?
Rode bieten.
Waarom begin je nou mee?
Nou ja, ik dacht dat is het enige wat niet leeft en niet wegkruipt.
Anyway, it works. That's all good.
Hoe gaat het?
Nou.
Yeah, he's alive. I'm gonna get some water for him. I think.
Die niet.
Ja, maar dan springen ze er ook heel snel uit, hè? Ja, tuurlijk leven.
Zweedse gehaktballen. Om ze op dat bord uit te stallen, zodat er extra bacteriën op gaan groeien.
Oh ja.
Dat betekent wel een stuk verantwoord.
Go get the whiskey. Go.
Zo.
Very good. Everyone's bringing me things that are alive.
Ja, ik doe nog steeds met ijs bij.
Je bent ook student?
Nee, al lang niet meer.
Is that alive, that thing? Is that alive? Get us more laboratory stuff. Turn that over so it's precious.
Ja, dit kan je trouwens ook nog frituren, hè?
Oh, nice.
Ja, maar Japanners.
I'm Simonter. What I'm doing, I'm making the collage, the still life.
So what do you have?
Some fruits, just for the visual aspect, the aesthetics.
Nee, dat is oké.
Okay.
So you have to ask the master first?
Yeah.
Dan de paling.
Maar blijft ze er heel lang meer staan? Ik vind het wel echt een beetje zielig.
Nee, maar juist om het minder zielig te maken is ijs goed. Want dan worden ze heel snel.
Je wilt geen joodlapjes aan.
Nee, joh.
Ook los.
Sorry.
Let's check again. I think they go off the table usually in the paintings, yeah? Okay. We're gonna start our lab. I probably need one of these jackets, damn it. We're gonna figure out how to isolate DNA from all this stuff, just from like household materials. First of all, we're doing something called hybrid DNA isolation. We need things that have DNA in them, and we need samples of them, and we need to put them in the blender. Put it in here, and then I'm gonna write apple.
Jullie hebben een kaartje voor mij lenen, want die staan.
What about sunscreen? Has sunscreen got DNA in it? What do you think?
I mean, yeah, just
Wash your fingers. Scrape something.
You're gonna get some hair? Dig it from the roots. Make sure you get the roots, like pull it out because the flesh is on the end, you know? You can also like go like this, like, yeah. That gets DNA flakes in there. We're working good together, huh?
Yeah.
I got one out.
Well, we'll get that one. Life. So even though it may still wiggle a bit, it is dead.
Hey, perfect. This is a laboratory. We mix salt and soap and contact lens solution, and we're gonna make DNA. Yeah. Of this, that's 8 cubic centimeters. You can put in a little more. It doesn't hurt.
And where can I see? Yeah.
So you don't have to measure too much. You know what I mean? Put a bunch of salt in. Oh, you do it. Well, let them say stop. Just tell them when to stop. Keep going. Don't listen to her. Okay, and then the next thing in there it says is twice as much water as there is samples.
okay.
So here you go. This is a super powered power stick. Now this is what I want us to do, and maybe we can I get you guys to do it? We have to stand up here, so stand with the crowd. But we also, we want everyone out here. Can you guys help us? Put it on and let's see what happens. I'll try this. Oh yes! We've got it! Stand back. I'm gonna electrocute myself. Okay. Yell it!
7 meatball, red beets, tulip,
Papaya! Cherry! Rhubarb! Yogurt! Arrowroot! Tomato! Pheasant head! Eggs! Lobster! Red berry! Apricot! Florocare! Ah! Oh! 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 Yuck! That's not a word. That's a word that fills in for the fact that there's no name for what you're feeling, right? It's like, right? Or shit. This is shit. It just means like, I don't know what it is, and we made up a word, it's a dirty word. Has this ever existed before on Earth? This specific milkshake? Okay, so we got something new here, right? We're scientists now. We're gonna actually isolate the DNA from something new. It's never existed on Earth before. A primeval soup of unnameability. And if we isolate the DNA from it, we can name it. Okay, so what does it take to do next? Everyone's scared now. It's gonna get groceries. Are you ready? Here's the option. We want to get this juice into these containers. Okay? So here we go. We've already established that this is something new on Earth, this mixture, right? No one has made something like this before. This is a new media, okay? And so it is possible, if you wanted to be a very contemporary new media artist, that you could sculpt with this and make a sculpture, a new media sculpture. And not only this, I do believe that if we sold it right, this would be a new media monoprint. Okay? This is a piece of contemporary art. This is how it's made now. It's not so bad. It's actually kind of clay-like. It's moldable. Yeah, there's an eye here. It's nice. Okay, but let's make some DNA out of this stuff. That's the juice. So here, about 1/6 of this liquid needs, you have to add an extra 1/6 of soap. So I'm gonna give you guys some soap. There are all these cells in here, but we want them to explode. So you're on the explosion team. You're on the explosion team. What it says is you add, no matter how much liquid you see, add about 1/6 more. Someone from the audience, you want to do it, right? I saw you getting involved. You ate the... You guys too, a little extra is better than not enough. Look at stuff. I like doing that. It's so mean, right? Oh, boil and bubble. They used to hang people here at the VOG for stuff like this, right? Now that the cells have exploded and probably the nucleuses have exploded too, the DNA is actually, it's in solution, but it's all wound up around all kinds of stuff, and we have to digest the DNA, which means like it comes more out into solution. When we start digesting it, it's delicate. Okay, so none of this stirring around. We're gonna have to be like lightly treating it, otherwise it'll break into tiny little pieces and it won't precipitate properly. So, anybody who wants a tube gets a tube. In fact, usually you're in charge of passing them to everyone, including the crowd. If you suck it all the way up into here, you can never use it again. But you know what? It doesn't matter. Like we're in the street, we can use them again if we want to. To get it out, you squeeze this button here. So this is just to get air, and this is up, and this is out. Right? And if you want extra out, you can do this. Ah! Right? Like that. But only fill it up. Okay. Wait. Let go of that one. Yeah, you got it. Oh yeah. Yeah. That's excellent. I think you learn faster than most adults. Good sign. So, you get to write down on your tube your own name for it. All right? Go for it. There you go. See, look at it. It took you 10 seconds. These guys took some. Right? There are a few things that digest DNA. They're the same thing that make meat softer. Okay? Papaya enzyme we take for digestion. Like I'm having a problem with my digestion. If you smash these up, you can add them as digestant to your test tubes, okay? But what really works is contact lens solution, okay? Contact lens solution is made to digest proteins that are stuck on your contact lenses, and that's how they clean them for your eyes, because you get all this glunk stuck on them, right? This deglunkifies them. You're gonna add some of this to your test tube, right? Like we've been doing. Ugh. And then, how does it say? It says to add a pinch, but we don't have a pinch. We have this bottle, so add some. That's the DNA kitchen way. You add some, right? Leave room, because we're going to put cold alcohol on the top, and then the DNA is supposed to go up into this, right?
Nou, heel voorzichtig zo.
All right, who needs And also don't zoom in, zoom all the way. I'm going to make a big one.
Hij zegt eerst dat hij hierop...
Zitten, ja. Hij weet wie Lilith is. Dat is de eerste vrouw van Adam.
Adam?
Adam, dan weet je wel, de eerste mensen op...
We're going to make a lot of DNA. Okay, so what do you think? Is the DNA ready to come out? We'll try it. We'll give it a whirl. If we pour some of this ethanol on the top of your test tube, the DNA will go up into it, like precipitate up into it. Why? Because the ethanol is floating on water and is cold. I don't know why. You tilt your tube and you just run the alcohol. Is this like something people do in bars? Make a black and tan, right? You make the alcohol go. Okay. You want it to float. This is the science part. What's the difference between ethanol, famous grouse, and some kind of crazy rum? It's like 80%. This is 80%, 90%, and 100%. You see these little white things coming to the top with bubbles?
Yeah.
That's the DNA. Okay, so come and try it. What you should see is something that looks a lot like sperm coming to the top between the alcohol and the juice. Should be like something spermy, like white, gooey, yucky, kind of icky stuff. As you're doing that, I also want you to tell me, after you added the alcohol, what name you wrote. These are, I want to know the names. Names of...
The name of the now we have.
Yeah, it's called shit acid soup.
Yeah.
You wrote it down
That's
already.
the DNA of the shit acid.
Yeah. It looks pretty good, right? I think we did okay. You know what I'll do for a picture of this? There you go.
Is it going to work? Such a big batch?
Why wouldn't it work? It's a big batch of the same small batch. Yeah, look at that. Looks like sperm coming up, right? Look at all that DNA.
Patrick is drinking all the rum.
Oh.
It's okay. That's what it's there for, actually.
Moi, c'est les trucs qui se
You gotta add the alcohol, Patrick.
T'as une partie de trucs plus dense et l'autre moins dense.
Pour it gently. You can use this too. First, we're gonna isolate the DNA. We got new tubes. We're gonna try a new tool. It's just like the other one. You suck up, and it'll suck something up, and then you squirt it somewhere else. That's all this does. So you press down, and then suck up, and then you press down, and you press all the way through, and it spits out. So.
Heel zuinig, hè? Je bent wel heel zuinig.
All the way through, like, yeah, spit out. Good.
Omdat iedereen naar de grond zit, dus dat is
And then you can take that tube home with your name on it. You can write it again. Well, that's nice. You got some good ones. Okay, how many genomes are in this DNA? Do I have to refer back? What would a scientist do with this DNA? Maybe. They might throw it away. This is a pain in the ass to figure anything out from. It's horrible, actually, right? Look at this. This is a lot of DNA from a lot of different genomes. It does kind of look like sperm, right? So what else can we do with this? Can we inject it into a plant? What do you think happens if you inject this into a plant? It might die, right? What if it lives? Will it be a mixture of all these and the plant? Something new? Could it be dangerous? Could be dangerous. What about injecting it into yourself? Same. Is it dangerous?
Yes, I think so.
Probably is. It doesn't look dangerous. You know, but I mean, I don't know, would you take a bunch of that and inject it into you? Right? The problem is if this DNA gets into the center of the nucleus, not just in your bloodstream, but into the center of a blood cell. And how does that happen? With a needle. Right, keep going. Don't burn your finger. Let me see. Good. What's that? That's the smallest needle. So small. It's actually not a needle yet, I can tell. Because I can't blow air through it, right? But if you break it here, that's a tiny needle. So what are we going to do? we're gonna take some of this DNA, we're gonna put it in this needle, and don't do this at home. No mouth pipetting, but we're gonna blow it down there. It tastes like lobster. You gotta find the nucleus of my cell.
Okay.
Okay, not too hard. Find one near the surface. Okay, and then...
En dan blow?
Yeah, well, let's get some more of this juice here. We're gonna really get in there. Okay.
Nou, we zullen zien. Oké.
Blow.
Ja?
Yeah.
Wow.
Ah! Okay, thank you. I think.
I think it's in.
So...
Oh!
What? I know. Oh yeah, definitely. We won't go into the testicles until you guys are gone. So what would be the difference if she blew this into my testicles? And my sperm would be different, and that would mean that I wouldn't just be different, but so would my kids, right? I think this is going on.
Yeah, I see something is happening.
Yeah, you feel it? You can, I'm changing. Well, anyway. Well, what else can we do with this DNA besides blowing it into me? Anybody else want it blown into them? Anybody want an injection into their testicles? What about a tree?
We can do a tree.
Should we do a tree? All right.
But we have to mark it as genetically modified then.
Oh, here, let's
Een sticker.
start this. What's that? Yeah. Tell me why. No, I'm interested.
No, because you're not going to change anything. Do it to yourself.
Yeah, do it to yourself, but not to something growing in the environment.
Truly.
Why not?
Why not? Because why you should leave it? No, it's part of life.
Yeah.
Thank you.
No. I actually, this is part of what this lab is about.
Yeah,
It's
this is
actually
incredible.
Trying to figure out, because it seems simple, but actually what she's saying is if I blow this into a tree and it takes part of the tree, and we don't know what the effect of all these going into a tree are. And we're just practicing maybe, you know, to make the tree supposedly make more money, right? But we don't know the long-term effect of what we do. I'm with you, just in case you couldn't tell. Yeah, I do it in a funny way, but this is the point. Wow!
Yeah.
Finally, someone said stop! It takes so long sometimes. I'm already deformed. Nobody stopped me. Well, okay. Believe it or not,
You have a free will, the tree doesn't.
that's right. Yeah, yeah, I asked for it. I got it. So, okay. That's our lab. I think we did it. Look at all that DNA. You see that? That's really a lot. And everybody got a little. I think all we have to do now is go home and think about what happened. But most important to me is that we name the unnameable. The weird thing about that, about making up a name for something that didn't exist before, that's what scientists do all the time. And you see, it's not that hard to make something new and name it. I don't know about copywriting it, you need a lawyer, you know, but it's actually, mystery is everywhere, right? This is mysterious stuff. It's not that hard. And they don't hang people for this anymore, they give them degrees anymore. Anyway, thank you.
Yeah,
Thank you. What a mess.
Finally, there's somebody saying stop. Finally, there's somebody really engaging in what we're doing here, and that's the process we want to get into. It's not biotechnology, and the very simple technology we're using here is not something only for experts. It's also to be educated to a general audience and to engage a general audience in and have them react on that. Adam wants to put this in an arts context by the expression bio-artist George Gesser gave to it, and also Jennifer Willett expressed that in her Inside Out project. If the military and agricultural and the medical industry is allowed to use living organisms and change living organisms for utilitarian reasons, why shouldn't an artist change organisms for art's sake? If you think about Craig Ventnor and the Human Genome Project, a few years ago they needed very, very heavy supercomputers to analyze the human genome. That process is now brought back, and you don't have to use that heavy computers anymore. So also that technology is democratizing. And there's a book called BioBazaar that tells about open sourcing biotechnology, taking away all the patents of biotechnology, using open licenses to use scientific knowledge that will eventually try to bring biotechnology to a very bricolage level where you can do it in your backyard or in your kitchen, whatever, for whatever use, for art, or maybe to process your food.
Yeah. I think that everyone can come up with some new idea again. They just mush it up. Okay. Let's get out of here. We ready to clean? Is that what's going on?
Yeah.
All right.
Where do we leave all those vegetables and fruits and?
We're gonna grab
things that are good.
We're bringing them home. We're gonna have dinner. But this one, he said, that is not okay.
Haag.org. Oké. Dankjewel. Ik hoop dat wij de volgende keer er zijn.
Nou, jullie zijn welkom.
Dat zetten jullie wel op die website,
Ja,
wanneer
natuurlijk,
jullie hier
natuurlijk.
komen.